Hello everyone. Uh, Now this is the lecture about the practical use of Swiss model. I have theoretically explained the usage of this server in my previous lecture. Now let us see how can we use it online. You will click on Start Modeling and then copy the sequence of your query protein and paste it. So it has become colorful. It means it is very easy for the Swiss model to process it for further analysis. You may give your project title protein 3. You may give your email address as well. Sorry, protein 3. And then we will go, as it is a server for the homology modeling. So first we will select the templates. But before selecting the template, searching for the template, you may select any of these two options. Like you may select the single target example or the multiple target example. In case of multiple target, our protein will be splitted in the form of two targets, but we are going to perform it using the single target. So prefer the single target sequence and then click on search for templates. And as I already told you in the theoretical lecture of Swiss model, that when the structure will be flowing in front, when the sequence of amino acids will be flowing in front of you, it means that the Swiss model is performing the blast analysis of your protein to be analyzed with the already reported proteins which are deposited in the PDB database until it will find the templates. And I have already found its templates, and because it takes some time, a little bit, maybe a little bit longer time. So you can see here it will show you the templates in this form. And as I mentioned earlier, that you have to select the template for building the model. Now and for selection of the template, you have to check the coverage, which is 92.59%. It is showing 92.59% identity with the fatty acid oxidation complex subunit alpha from E. coli, and its GMQE value is 1, which is also the reliable value, and it is also the structure of this template has been predicted using the X-ray crystallography. So the structure which we will build using this template will be having a high confidence, will give us high confidence, because that structure or model will be predicted using an already experimentally predicted structure. Now click on the build model. So it has taken lesser time because I have already built the model using the same template. Otherwise it takes a little bit longer time. Now you can see the 3D structure of your protein, and here is the GMQE score, which is 0.94, and next, and the Q mean score is 0.92. You can see your protein structure in any format, like in tube, in lines, in ball and stick, but the preferred one is cartoon, which we normally use for the presentation in the manuscript. And then you may also click on the play button. This will start rotating your protein, and you may select any of the angle from which you want to take its pic in order to represent in your manuscript. You may also move it with the help of a cursor. So, and you may also check the z-scores. As I already told you, this is the graphical representation which is showing that our model is lying in the gray region, but or the darker region, but actually it will be much preferred, would be the structure would be much preferred for the docking analysis or any other analysis if the structure falls in this darker region. So but here our model falls beyond this value. Therefore, the z-score value is minus 0.57, and I told you that if the z-score value is below the minus 0.4, then the atoms and the bonds of the predicted structure are not reliable enough to process to use it for any other analysis. Like most commonly we use it for the docking analysis, or you may use it for the conserved domain prediction analysis, etc. So this was all about the practical use of Swiss model. I hope you would be able to perform it properly in future for your article preparation. Thank you so much.